Good evening, everyone. Good evening to all the students. So today is day 4 of our entire session, right? हम already 3 sessions कर चुके हैं related to the biochemistry, and today is the 4th session for your biochemistry unit. So if you want, you can watch the previous lectures as well for learning and understanding the questions and everything. Please confirm whether the audio and the video is clear to everyone or not. If yes, let's begin with the class. Good evening, Bushra. Good evening, Ritika. Okay. Is the voice better now? Yes, is the voice better now? Actually, I'm not keeping well also today a bit. That's another reason for low sound. So you have to bear with it a bit. Otherwise, just check, is it better than the previous time? Yes, is it better now? Yes, is the audio better? Okay, let's begin with the class. So if you're someone who's preparing for the examination with a good proper serious approach, we have a Lakshya batch ongoing completely free of cost for you, in which on daily basis you get the free PDFs, free test and everything for your preparation. Then in addition, we have a success batch ongoing. We have recorded batches, full courses, gate excel modules in the soft copy format, and we have hard copy notes as well available. So if you wish to place order for any of these things, you have the WhatsApp number in front of you. You can contact us here and place your orders. Right, let's begin with the class. This is the first question for you today. This is a question from biochemistry and cell biology both. The question is, the signal sequence direct the proteins to correct intracellular locations, right? When we learn about the protein translocation, in the protein translocation you have, let's say, one protein, it need to be translocated to, let's say, lysosome. Yes, free tests are available. Many every day free test conduct karaya jata hai, so you can join in that as well. So let's say you have a protein that need to be transported to lysosome. So mano 6 phosphate उसका टैग होगा. अगर वो न्यूक्लियस के पास जाना है तो एक टैग होगा. अगर उसको केडी, अगर उसके एंडोप्लाज्मिक रेटिकुलम के पास जाना है तो उसका KDEL सीक्वेंस होगा. अगर उसे किसी और जगह पर जाना है सो एक स्पेसिफिक ओरिएंटेशन होती है. क्वेश्चन में पूछा है दैट विच ऑफ द फॉलोइंग सीक्वेंसेस इज टिपिकली यूज्ड टू इंपोर्ट द प्रोटीन इनटू द न्यूक्लियस. इसी सेगमेंट के अंदर से प्रोटीन सॉर्टिंग के कॉन्सेप्ट से पूछा गया है कौन सा स्पेसिफिक प्रोटीन हेल्प करेगा फॉर द इंपोर्टिंग इनटू द न्यूक्लियस ऑल टुगेदर. ट्राई द क्वेश्चन एवरीवन, देन विल डू इट टुगेदर. यस, what should be the answer for this? अगर किसी specific sequence को import होना है into the nucleus, so what should be the sequence, or what should be the tagged sequence altogether? SKL is for your peroxisomes. KDEL is for your endoplasmic reticulum. Yes, first is the answer for this particular question. Why? Because जब एक पर्टिकुलर प्रोटीन को न्यूक्लियस में इंपोर्ट होना है, सो इट कुड रिक्वायर हायर कॉन्सेंट्रेशन ऑफ लाइसिन एंड like majorly lysines highest concentration need to be therein. So अगर हम चारों ऑप्शन को ऑब्जर्व करेंगे, फर्स्ट इज द करेक्ट आंसर. रीजन भी जो लाइसिन है वो हाईएस्ट कॉन्सेंट्रेशन में फर्स्ट में ही रिप्रेजेंटेड है. सो एज अ रिजल्ट ए इज द आंसर फॉर द क्वेश्चन. राइट, इजी एंड सिंपल क्वेश्चन. आई होप इट्स क्लियर. सो सिग्नल सीक्वेंस डायरेक्ट द प्रोटीन्स. सो कौन से जगह पर वो मेजरली इम्पोर्ट होना, मतलब अगर प्रोटीन में इम्पोर्ट होना है सो क्या मेजर वहाँ पर सीक्वेंस होना चाहिए? इट शुड बी लाइसिन. राइट, सो वेरी इजी. ए इज द आंसर फॉर दिस. सो रिमेंबर दिस पर्टिकुलर थिंग कि प्रोटीन enters into the nucleus via the nuclear localization signal. और एक छोटा सा जो nuclear localization signal होता है, it is a short stretch of the basic amino acids which is lysine, arginine specifically, right? So classic example is this, which matches with option number A, right? K is what? K is nothing but your single letter code for your lysine, right? So remember about this thing as well. Then just a second. In addition to that, we also have the important proteins that recognizes this sequence and transport the protein into the nucleus via the nuclear pores. So यहाँ पर ये question हम specifically cell biology में ही रखेंगे because ये nuclear transportation को भी cater करता है to some extent. So keep a note of that as well, right? So in short, NLS is the cluster of basic residues, specifically lysine and also arginine, that directs the protein to the न्यूक्लियस. इसीलिए ऑप्शन नंबर 1 आंसर होगा. सो रिमेंबर अबाउट दिस इंटायर इंफॉर्मेशन एज वेल कि क्वेश्चन का आंसर ए है बट आंसर ए क्यों है दैट इज ऑल्सो इम्पॉर्टेंट. राइट, सो कीप अ नोट ऑफ दिस एज वेल. ओके, नेक्स्ट क्वेश्चन. द यूनिट ऑफ मोलार एक्सटिंक्शन कोफिशिएंट आर. जो मोलार एक्सटिंक्शन कोफिशिएंट एक स्पेसिफिक एंटाइटी होती है हमारे बायोकेमिस्ट्री में, उसकी यूनिट क्या होती है? द यूनिट ऑफ मोलार एक्सटिंक्शन कोफिशिएंट आर विच ऑफ द फॉलोइंग? ट्राई दिस एवरीवन, गिव इट अ ट्राई. क्या आंसर होगा इसका? ट्राई इट आउट. एंड आई एम सॉरी फॉर लो वॉइस टुडे. आई एम नॉट कीपिंग वेल and I still wanted to make sure that I am having at least a short session so that you can solve out few questions here today. Done everyone? This question, mark the answer for this question now. The unit of molar extinction coefficient R. Everyone mark the answer. Done? What is the unit of your molar extinction coefficient? It's very simple question. So it is option number 1. That is, over here the concept of your pure Lambert law is being asked in. A is absorbance, E is your molar extinction coefficient, C is your concentration and L is your path length. So यहाँ पर जो मेरा entire coefficient होता है, that is for your the entire unit for the same is liter per mole per centimeter, right? So keep a note of this particular thing. Very easy, very important. कभी कभी एग्जाम के अंदर आपको स्पेसिफिक कुछ इंफॉर्मेशन देकर एब्जॉर्बेंस कैलकुलेट करने को भी बोली जाती है, राइट? सो नोट, कीप अ नोट ऑफ दिस एज वेल. आई होप इट्स क्लियर. It's no, we'll have the questions discussed at least for some time, don't worry. Next question. The amount of hemoglobin in the blood is one of the most important health markers. Statements are given to us regarding the yeah, so the question is, the amount of hemoglobin in the blood is one of the most important health markers. Following statements are made regarding the hemoglobin degradation when older red blood cells are destroyed by the tissue macrophages. A, B, C and D. Fourth options are therein. We need to find out which of the combination is the correct one. The question is asking us the information related to the hemoglobin degradation. Try this, easy. Mark this, everyone. The amount of hemoglobin in the blood is one of the important health markers. The statements are given to us regarding the hemoglobin degradation when the older red blood cells are destroyed by the tissue macrophages. The question is asking us about the correct combination of statements. Try this, everyone. The hemoglobin protein of the globin part of the hemoglobin is split off and the heme is converted first to bilirubin by the action of heme oxygenase. Then you have the globulin protein of the hemoglobin is split off and heme is converted first to biliverdin by the action of heme oxygenase. Then biliverdin, then carbon monoxide is formed in the process. Then nitric oxide is formed in the process. Mark the answer for this question, everyone. Done, everyone? Yes. Ritika, I'm not feeling well. How should I shout in the class today? I'm not keeping well today. What should I do to shout? I'm trying my level best to take as many questions as possible. The amount of hemoglobin in the blood is one of the most important health marker. The following statements are made regarding the hemoglobin degradation when older red blood cells are destroyed by the tissue macrophages. So question is asking us which of the following combination represents all the correct statements. So out of carbon monoxide and nitric oxide, the carbon monoxide is formed and not the nitric oxide, right? So you can eliminate option number D. Then between the first and the second one, that first is the do we have the biliverdin being formed in or the bilirubin being formed in? So it is your biliverdin formed and then the bilirubin, right? When you learn about the Ritika, you can please leave the class if it is that difficult for you. I'm really, really sorry, bache. I'm already having fever. I'm already not keeping well right now. How should I do it? You tell me, what should I do? I cannot speak louder than this today. Either I should end the class for you. When the older red blood cells are broken down by the macrophages, the hemoglobin is first split off into the globulin part and the heme part. Then the in the heme oxygenase, like the heme oxygenase enzyme that we have, the heme, it acts on the heme part to convert it first into the biliverdin and then the bilirubin. Then during this process your carbon monoxide is released and not nitric oxide, right? So keep a note of this. First it is your biliverdin that is formed and not bilirubin. Then in the hemoglobin, the globulin part and the heme part are split off and you have the formation of your carbon monoxide and not carbon dioxide, right? So what should be the answer for the question? The answer for the question is option number C. B and C is the correct answer and A and D are the wrong options, right? So answer for this is option number 2. I hope it's clear. Next question, and I'm really sorry, I'm not saying it in any rude way, but I'm not keeping well, bache. I'm really sorry for this. What should I do to increase my voice? I have kept a mic and everything is there. If it is not doing fine, just try to accommodate for one day at least. You're continuously saying the same thing. Is it that not audible to anyone in the class? Yes, Bhargavi is there and other Megha is also there. Is it that problematic, bache? Just let me, I can end the class if it is that bad or if it is that problematic for everyone. Just let me know. I'm really sorry, but I'm also a human, right? I can also fall ill. Try this question if I am audible to everyone. The Ramachandran plot graphically shows which combination of the torsional angles phi and psi of the amino acid residues in a peptide are possible. Examination of the plot below shows that only certain regions of the conformational spaces are permissible. Question is asking why all the phi and psi angles are not possible. Question is asking us why do we have that all the phi and the psi angles are not, all the theoretical combination of the phi and the psi angles are not possible in. Try this question, everyone. And this is the last question. I'm just not keeping well right now. I won't be able to continue. We'll have this is the last question. I'm not feeling well right now. Mark the answer for this question quickly. Done, everyone? Okay. Question is why all the theoretical angles of phi and psi are not possible in the Ramachandran plot? The answer for this is, see, look into the entire reason over here. So no, the two atoms cannot occupy the same space. Reason being, you have the restriction, like this restriction is because of the conformational space and everything. So because your two atoms cannot occupy, reason being the two atoms have a different conformation or a different um, you have atom A and atom B, let's say. So let's say this is the conformation, or this is the structure of A, and this is the structure of B. So these two cause hindrance in each of its structure. So it can cause hindrance. So that's the reason why all the phi and psi angles are not possible, because the two atoms cannot occupy the same space altogether, right? So A is the answer for this particular question. Simple and easy, not difficult. You just have to remember this information, right? And I'm really sorry, I'm just not able, I won't be able to continue. I'll have the class tomorrow, but right now I'm not able to sit and like continue the questions. You all can check the recorded part, and I'll take up the live class tomorrow. I'm really sorry, but not able to, I won't be able to continue right now. We'll have it tomorrow. Okay, so you can watch the playlist for the other videos, and we'll have the discussion in the next class. Bye everyone, take care.